Transcriptomics 3 is a course dedicated to advanced methods of analysis for transcriptomic data that will allow us to find meaningful patterns in data, especially complex patterns that are typically present in big datasets. In this video, we will speak about unsupervised learning methods, such as clustering. Complex patterns that are present in large datasets are hard to find manually. These types of data show nonlinear dependencies and contain noise that make it hard to find statistically significant differences. This is where we can turn to automated learning, which is also known as machine learning. Here, we can train an algorithm or a machine to perform tasks with data, such as mining for patterns. Machine learning can be separated into two main categories of methods, both unsupervised and supervised machine learning. Unsupervised machine learning focuses on detection of patterns in data. The main idea is to apply techniques that are automated and rely on various assumptions about data to learn from it. The second main category is the supervised machine learning part. These methods rely on availability of training data, which is data that has been labeled. Once a data set of labeled data is accumulated, it can be used to train a model that will be able to apply this knowledge to analyze new data, for example, by predicting its class. In this course, we will start exploring different methods for identifying groups of samples without prior knowledge. This process is called clustering. And then we'll examine methods for developing classification models from known samples in order to classify data present that is in new samples. In this video, we will speak about clustering. Clustering gene expression data is one of the most useful techniques that you can use when analyzing high-throughput molecular data. Not only can it help you find patterns in the data that you did not know existed, but it can also be useful for identifying outliers, incorrectly annotated samples, and other issues that are hard to spot with traditional methods. With unsupervised machine learning methods, we'll start with a dataset, which is typically a table of elements, such as these shapes. We can use a clustering method, for example hierarchical clustering, which is a method that seeks to identify groups based on their similarities. We can also use a dimensionality reduction method, such as principal component analysis, that will use data variance to assign samples to groups of elements that are present. In some cases, we can use results to draw other conclusions about the data within such a structure. In this case, groups based on the shape of elements within the group. This is the new information that this technique will generate. Principal component analysis uses dimensionality reduction to show us what the data variability will look like. In PCA, we use the approach of principal components that explain variance in our data in the best possible way. Here you see data with low variance. The samples are grouped close together. And then here's a sample with higher variance. However, this variance is not well explained by the x and y axes, but you could draw a line through the original space that shows the maximum variance of this dataset. This vector through the multidimensional space maximizes the variance of the data and is called a principal component. And just like with the previous example, we can add other principal components, such as PC2 or even PC3, and thus visualize our data by projecting it into a space of 2 or 3 axes that explain some percentage of data variance. When we select the principal components that best explain the most variance of our data, we can project all of our samples into the space and then visualize the full dataset. This technique is called principal component analysis, and it can be very helpful in the initial stages of our analysis. We can liken PCA to a photographer trying to find the best angle for a picture that will capture as much detail as possible, while showing us the most information about the group as a whole. We would typically use this method to visualize and explore our data. When using this multivariate analysis method, it's important to play around with the data using normalization and filtering, while paying attention to outliers for the best possible interpretation. But PCA does not identify groups or their boundaries, and that's when we have to turn to clustering. There are many types of clustering, and hierarchical is one of the most popular approaches because it allows clustering on different levels. Let's say we have our samples organized in some space. So imagine a PCA 2D plot where samples are positioned in some perspective that best represent the data variability. The algorithm would find the two closest objects in this projection and then link them together. This is a cluster. Then, we would find the next two closest group of two, where any two linked objects are considered as a single object, and their location is the center of that cluster. The algorithm will run until all the objects are assigned to a cluster, with all the objects in it. To run clustering on the T-bioinfo platform, you'll need to upload the table of expression that we generated from analyzing the cell line data. This data can be downloaded directly from the lesson, or from a demo pipeline in the cell line RNA-seq demo. The clustering methods are located in the data mining section of the platform. Hierarchical clustering is an example of an agglomerative, or bottom-up clustering method that starts with attributing each sample to a separate cluster. It then finds the pair of most similar samples and unites them into one cluster. This procedure is repeated until coming into one cluster that contains all the samples. The pipeline will take the output, perform clustering, and then put out the result in several formats that we will discuss in a little bit. But what are the parameters? The linkage parameter determines the definition for measuring inter-cluster difference. Single sets the definition as the distance between the two nearest points in two clusters, whereas complete defines the distance as that between the two farthest points. The two Ward methods are variations of Ward's minimum variance method. The distance parameter specifies the distance metric to use in measuring distance between data points. These can be Euclidean, Manhattan, maximum, or Canberra. The cluster's expected count parameter sets the cutoff height to the one of the results in a given number of clusters. Do transpose allows the user to transpose the input matrix to set whether clustered objects, or samples, are represented by rows or columns of the input table. We can use this clustering to cluster objects, or samples, or even features, which are genes. What is cluster linkage? Well, there are several types. The average linkage method finds all possible pairwise distances for points belonging to two different clusters, and then calculates this average. This method is less affected by outliers than the others. Single linkage uses the minimum difference to determine clustering. Remember that this method is also known as nearest neighbor clustering. The distance between two groups is defined as the distance between their two closest neighbors, and more formally, it finds the minimum possible distance between points belonging to two different clusters. This method often yields clusters which individuals are added sequentially to a single group. The complete linkage method uses the maximum, and is also known as the furthest neighbor, or maximum method. This method defines the distance between two groups as the distance between their furthest apart members. More formally, it finds the maximum possible distance between points belonging to two different clusters. This method usually yields clusters that are well separated, as well as compact. The centroid method looks for the center of each cluster, and then calculates the distance between the centroids of those two clusters. In addition to our decision of linkage criterion, for hierarchical clustering, we also need to indicate the distance metric. If Euclidean distance is chosen, then observations with high values of features will be clustered together. The same holds true for observations with low values of features. Remember that with Manhattan distance, xi and yi are x and y coordinates of the ith observation. Canberra is a weighted version of the Manhattan. The choice of distance measures is important, as it has a strong influence on the clustering results. For most common clustering software, the default distance measure is the Euclidean distance. If Euclidean distance is chosen, then observations with high values of features will be clustered together, and the same holds true for observations with low values of features. The hierarchical clustering algorithm is a process, and in this process, the computer uses the input file where columns are objects and rows are features. The algorithm assigns each group to a separate cluster, and then evaluates all pairwise distances between those clusters using a chosen distance metric. It then constructs a distance matrix using the distance values, and then finds a pair of clusters with the shortest distance. It merges them into a single cluster, and then repeats from step 2 until we end up with a separate cluster for all the samples, or objects. With the k-means pipeline, we inputted a table of expression in .txt format, and after running the algorithm, we were able to observe 3 different things. The first was that the k-means clustering does not do a great job of clustering. Next, we found that Basel was split into 2 different clusters, with cloud and low in one and normal-like in the other. And lastly, we found that luminal was split into 2 different clusters. Our output was a .txt file as well, with a cluster ID. The difference between hclust and k-means is that hclust has an output such as a dendrogram that gives you the structure of your data, whereas k-means just gives you the number of each cluster in each sample. This is a good example on how different levels or heights might have different numbers of clusters or subclusters. As we go towards the top, there are less clusters and the separation is going to be stronger. A good way to test clustering models are to actually compare phenotypical data with the unsupervised classifications that we create. Clustering is also widely used in data visualization to organize the patterns that we get into heat maps so that we can visually see groups of samples and genes. In this section, we also spoke about different methods for identifying groups of samples without prior knowledge. We saw how gene expression clustering is one of the most useful techniques that you can use when analyzing gene expression data. Not only can it help find patterns in the data that you did not know existed, but it can also be useful in identifying outliers. We spoke about several different methods for clustering, such as hierarchical and k-means clustering, and we learned how to perform these analyses on the tBioInfo platform. In our next session, we will speak about supervised machine learning as well as classification.